section, we'll take a look at a specific example of catabolism, carbohydrate catabolism. So let's see what happens if we're eating the crust first. In our first step of carbohydrate metabolism, and to eat our pizza crust, we have to break down the complex material, the flour, that's the primary material in that crust. Starch is a polymer of glucose. It's essentially a complex structure made of repeating molecules of glucose. We have glycogen that can also be found as a storage carbohydrate or polysaccharide in animals that's also made up of repeating units of glucose. Now, starch in the form of the pizza crust has to be broken down, and we're going to use an enzyme called amylase to break those glycosidic bonds between the glucose molecules and release those glucose individual molecules for the next step in our process of catabolism. It's called glycolysis. Now, for our class, I'm not going to ask that you memorize all of the steps of glycolysis. You'll not need to memorize the names of all the chemicals, enzymes, the step-by-step process. A more appropriate class for that kind of task and requirement would be a biochemistry class or a cell biology class. In our course, it's more important for us to understand what is important about each particular metabolic process. What's the important outcome? What are the important events? For glycolysis, we need to understand that this is not an oxygen-dependent process. That glycolysis can occur in an oxygen-free environment, that we're going to find substrate phosphorylation will occur, and we'll explain that in more detail as we go through the key points or the highlights of glycolysis. We're going to take a 6-carbon molecule in the form of glucose and break it down into 2 3-carbon molecules. In other words, we're going to take that 6-carbon molecule and cut it in half. In the process of separating the 2 3-carbon components of that original 6-carbon glucose, we're going to transfer some electrons and some hydrogens. We're going to capture those electron and hydrogen elements or portions of the molecule into something called NADH. We're also going to capture some energy in the process of substrate level phosphorylation and create some energy containing or energy potentially containing molecules called ATP. So we just need to remember the key steps and the important outcome for glycolysis. We'll review this one more time after we go through each step. We will start glycolysis by taking 1 molecule of glucose, adding a phosphate group from ATP to one end, and a second phosphate group from a second ATP molecule, and attach a second phosphate group to the opposite end of the 6-carbon molecule of glucose. We need to attach those phosphate groups because it's very much like adding a red light or a sparkler to the molecule. Identifies some key components or structures of the molecule for enzymes to attach and essentially facilitate the breaking of the bonds between essentially the 2 3-carbon components of glucose. We essentially want to cut that 6-carbon glucose right in half so that we can eventually make 2 3-carbon molecules. So we start with glucose. We add 1 phosphate group to one end that gives us glucose 6-phosphate. We add the phosphate group to the other end, and now we have fructose 1,6-diphosphate. Keep in mind that glucose and fructose are isomers of one another. They're both 6 carbon molecules. They both have 12 hydrogens, and they both have 6 oxygens. They're just arranged in a different pattern. The next series of steps in glycolysis are some of the most important. We're going to take fructose 1,6-diphosphate and with the use of a key enzyme, cleave or cut this 6 carbon molecule into 2 3 carbon molecules. Initially, the 2 3 carbon components that are produced are not identical. One is dihydroxyacetone phosphate, the other is glyceraldehyde 3-phosphate. But there is an enzyme that will modify the dihydroxyacetone phosphate into a second molecule of glyceraldehyde 3-phosphate. We now essentially have twins. We have 2 identical molecules of glyceraldehyde 3-phosphate. Now we need to modify this a little bit more. We need to attach a second phosphate group to the opposite end of each glyceraldehyde 3-phosphate. We're going to do that by plucking a phosphate group from another source of phosphates, like an ATP, and we're also going to reduce a molecule of NAD and create some NADH. We're going to make 2 NADHs in this process, and we're going to phosphorylate or add a phosphate group to the 2 glyceraldehyde 3-phosphates to create 2 molecules of 1,3-diphosphoglyceric acid. The next few steps of the reactions for glycolysis get us toward the end molecule that's important, which is pyruvic acid. Ideally, summarizing glycolysis in one sentence would simply say we're going to take 1 6-carbon molecule of glucose and make 2 3-carbon molecules of pyruvic acid. Now to get to that point, we now have the 1,3-diphosphoglyceric acid molecules, 3-carbon molecules with a phosphate group on either end. We're going to dephosphorylate into 3-phosphoglyceric acid. Dephosphorylation means we're removing a phosphate group. We remove a phosphate group from one end of the molecule. The next step, we're going to move the phosphate group to the middle of the 3-carbon molecule and rearrange the grouping. Also, we're going to remove a molecule of water, or dehydrate, and create 2 molecules of phosphoenolpyruvate. Now, phosphoenolpyruvate still has a phosphate group attached to it. We have to remove that. We're going to remove the phosphate group from the phosphoenolpyruvate, and that leaves us with 2 pyruvic acid molecules. Now, the dephosphorylation steps. We're removing those phosphate groups from the organic molecules, putting the phosphate group on ADP to create some ATP, or adenosine diphosphate, into adenosine triphosphate. We do that 2 times, so we make essentially 4 molecules of ATP in this bottom half of glycolysis. But if we go to the top half, remember we used 2 molecules of ATP for the original sources of phosphate groups. So our net yield for glycolysis would be 2 molecules of ATP. Let's summarize again the importance of glycolysis that you need to know for our class. 6-carbon glucose is going to be cut into 2 3-carbon molecules of pyruvic acid, which can also be called pyruvate depending upon the ionization state. We're going to create 2 molecules of ATP, net yield, from the process of substrate level phosphorylation, taking glucose and attaching phosphate groups to it, and then moving those phosphate groups around as we modify the 6-carbon molecule of glucose into the 2 3-carbon molecule pyruvic acids, and also using those phosphate groups to create some ATP net yield. We're also going to create 2 molecules of NADH. And in fact, we're going to find it's the NADH that's the more useful of the 2 molecules. We actually have a potential net yield from each NADH of 3 ATPs per NADH. So that really is the more high energy molecule, potentially, than the single ATP.